Good evening, everyone. Today we'll be discussing about the discussion week 7 of the course Introduction to Cell Biology. And in this lecture, we'll be discussing about the endomembrane system of the cell and the different components, including endoplasmic reticulum, Golgi bodies, lysosomes, and the vacuoles that are the consistent part of the endomembrane system. Okay, so I am Rishabh from IIT Kanpur and let's start the lecture. So the first question for today is, which of the following statements about the endomembrane system is true? So what can be the correct answer? So the correct answer is option 4. That means both 2 and 3 options are correct. So what is exactly the endomembrane system? So the endomembrane system actually comprises about more than 40% of the membranous part of the cell. Okay, so it starts with the endoplasmic reticulum that is connected to the nuclear envelope. Okay, so endomembrane system starts with endoplasmic reticulum where the protein synthesis occurs. Okay, then from here the proteins move to Golgi bodies through vesicles, and from Golgi bodies the different proteins move to either plasma membrane or secreted or lysosome. Okay, so likewise, this is the flow of the protein movement from the endoplasmic reticulum where it is translated and to the outside or the plasma membrane or the lysosome where the protein is actually destined. Okay, so now let's see what's in the diagram. So this is the nucleus. Here is the nuclear membrane. The endoplasmic reticulum is continuous with the nuclear envelope, and from here the protein molecules or the cargos are actually shifted in the form of vesicles to the Golgi bodies. Hmm. Okay, so from the Golgi bodies, the cargo is actually transported in the form of vesicles again to the plasma membrane or to the outside the cell, that is secreted. Okay, so this was the function of the endomembrane system. So any doubts in this question? If not, then we'll move on to the next one. So the endoplasmic reticulum membrane, which is associated with ribosomes, is called. So what can be the correct answer? So the correct answer is option 1, that is rough endoplasmic reticulum. So what happens is when so for example this is the nucleus and this is the nuclear membrane. Okay, and from the nuclear membrane this is a continuous membranous network. Okay, so now this is called as the endoplasmic reticulum. Now, when what happens is there are two kinds of ribosomes. Okay, so the one type of ribosomes are that is free in the cytoplasm, and the other ones are attached to ER. Okay, so the ones that are attached to the ER membrane give it a rough appearance in terms of looking. So, this rough appearance given by the ribosomes on the endoplasmic reticulum gave it a name as rough endoplasmic reticulum. Okay, the other one that we have is the smooth endoplasmic reticulum, that doesn't have any ribosomes attached to it. Okay, and it gives a smooth appearance. Okay, rough endoplasmic reticulum has the function of ribosome is translation. Okay, so if the ribosomes are attached to the ER, that gives us the idea that the proteins that need to be secreted or present on the plasma membrane are present on or will be produced by the ribosomes that are will be produced by the ribosomes present on the endoplasmic reticulum membrane. Okay, so on the this thing, electron micrograph, as you can see, because the presence of the ribosome gives a rough appearance on the endoplasmic reticulum membrane, so that's why this is called as a rough endoplasmic reticulum. And here on the left hand side, where the smooth endoplasmic reticulum is present, if the ribosomes are not attached to the surface or the membrane of the endoplasmic reticulum. Hence, they are called as smooth endoplasmic reticulum. Okay. So, any doubts in this particular question? If not, then we'll move on to the next question. How does the endoplasmic reticulum assist in protein folding? So, what can be the correct answer? So, the correct answer is option 2. That means it houses the molecular chaperone that assists in protein folding. Okay, so this is the case. For example, what happens is the ribosomes. So, this is the mRNA, and these are the ribosomes. Okay, so when the ribosome is actually translating on the mRNA, the the proteins are actually moving in the endoplasmic reticulum, and there are some chaperonic proteins that are present in the lumen of the endoplasmic reticulum. They are present in the lumen of the endoplasmic reticulum. These proteins actually assist in protein folding, giving a conformational change to the protein. Okay, so this final conformation lead to their function. Okay, so this is how the endoplasmic reticulum actually helps in protein folding. Okay, so any doubts in this particular question? If not, then we'll move on to the next question. Which organelle is involved in the synthesis of membrane bound proteins? So, what can be the correct answer? So, the correct answer is rough endoplasmic reticulum is the rough endoplasmic reticulum. Okay, so how does it do is, so as I told you before, ribosomes are basically of two types. First is free, that are present in cytoplasm, and the other ones are bound to endoplasmic reticulum. Okay, making it a rough endoplasmic reticulum. So the ribosomes that are free, what will they do? Okay, so they will translate those proteins that are present in nucleus, cytoplasm, mitochondria. Okay, and the ones that are bound to ER will produce the proteins that are present on the plasma membrane, Golgi body, secretory proteins present in lysosomes, and peroxisomes. Okay, so this was the case. So when ribosomes will be present bound to the ER membrane, they will produce proteins that will reach to the plasma membrane. Okay, so they will directly go to the membrane and they will stick to the membrane. Okay, so this is how the ER, rough endoplasmic reticulum, helps in production of proteins that are bound to the membrane. Okay, so any doubts in this particular case? Let's move on to the next question. Rough endoplasmic reticulum is abundant in cells that, what can be the correct answer? So the correct answer is option 2. That means that secrete proteins. Okay. So here we need to understand another concept, that is the concept of signal peptide. Okay. So what is actually a signal peptide? So if a protein, for example, is destined to nucleus, so there will be a signal needed to reach this protein to that nucleus. Okay, so what happens is there is a short sequence of proteins, short sequence of amino acid present towards the end terminal of the protein. Okay, so what will happen? This will direct the protein to the specific location. Likewise, if the signal peptide is there, that means it the protein will go to mitochondria or plastid or plasma membrane and so on and so forth. Okay, so this is the major role of the signal peptide here in the cell. Okay, so now what happens is whenever uh, there is any secretory protein there, so there is actually a particular movement of the cargoes inside the cell. So, for example, as we told you, in the ribosomes that are present in the cytoplasm, these produce proteins that move to nucleus to and fro, the plastids, mitochondria, peroxisomes, and the endoplasmic reticulum. Okay, from from the endoplasmic reticulum, the proteins that are produced move to peroxisomes, Golgi bodies. From Golgi, it moves to late endosome, lysosome, early endosome, exterior, or through the secretory vesicles. Okay, so this is how the different proteins are distributed among different organelles in a particular cell. Okay, so let's move on to the another concept, that is how endoplasmic reticulum helps in protein folding. Okay, so what happens is, as I told you, there is a concept of signal peptide that actually gives us the signal that where the protein will be present. Okay, so the way where the protein will be present in which organelle. Okay, so what happens is, this is the mRNA. mRNA would be present only in the cytoplasm. So in the cytoplasm, the ribosome gets attached to the ribosome binding site of the mRNA, and then what happens? It will produce the proteins. The initial protein produced will have the signal peptide. This signal peptide will be recognized by a protein called as signal recognition particle, or the SRP. Okay, so this SRP is actually very important, where it directs the protein to be produced by rough endoplasmic reticulum. Okay, this SRP is actually recognized by a receptor or the translocation complex present on the rough endoplasmic reticulum membrane. Okay, now this when this is recognized, what will happen? The SRP will direct the protein to be synthesized within these particular endoplasmic reticulum, and it will be translocated in such a way that the signal peptide is attached to the translocation complex and the other protein moves to the inner lumen of the endoplasmic reticulum. Okay, so this protein will slowly get synthesized by the ribosomes, and it will start moving in the endoplasmic reticulum membrane through this translocation complex. Once more than half of the protein moves to the through the translocation complex, this signal peptide is cleaved. And after cleaving, this particular protein goes inside, and inside there are certain chaperone molecules present. Okay, so chaperone molecules actually assist in the folding of the protein. And here the protein, once the protein gets folded, it will achieve a final conformation that will actually lead its way to the Golgi bodies. Okay, so this is how the endoplasmic reticulum uh, takes place. They helps in protein folding. Okay, here two main chaperone proteins are present. That is carnitine and calreticulin. So both of these require calcium ions for their function. Okay, so this is how the endoplasmic reticulum actually helps in the protein folding. Okay, so let's move on to how the endoplasmic reticulum actually helps. Okay, so there was some. Okay, so carbohydrates are also actually carbohydrate. The main site of synthesis of carbohydrate is not in RER and SER. Okay, so this is basically the carbohydrates are mainly synthesized in the cytoplasm. So, in this particular case, when the particular protein needs to be folded, the enzymes are actually translocated inside the RER where they are synthesized. Okay, so likewise, whenever there is a need of something, then the cell actually translocates that particular enzyme to the compartment. So the major site is not RER, it's the cytoplasm for the carbohydrate production. But because it's needed here, that's why the major enzymes are present here. Okay, so now what happens is after like particular thing is added to this protein, then it is actually recognized by the chaperones. Okay, so now what happens is this is actually a schematic of how it's added to this particular protein molecule. Okay, so when the protein is fully folded and it's actually being attached to the particular oligosaccharide in the proper numbers, then it's just translocated to the Golgi. Okay, this is a schematic where it one by one all the molecules are added. Okay, so what happens is first the production starts from the cytosol site, and then but when this particular mannose proteins are added, this is recognized by one of the enzyme called as flippase. This particular enzyme actually flips the outside part to the inside part of the endoplasmic reticulum. Okay, so this is how it helps in the like production of more number of oligosaccharides inside the endoplasmic reticulum neuron. And then once the oligosaccharide is formed, it's transported or it's attached to the asparagine residue that is on the protein molecule. And then what happens after this attachment, there is a chaperone called as calnexin. Okay, this recognizes this particular oligosaccharide, and what happens if the required number of glucose are not present. Okay, so it will what will happen, it will actually help in glucose trimming or glucose addition. Okay, so what happens is once the protein needs to be folded, it needs to remove these particular glucose molecules. As you can see here, there are three glucose molecules, and for the protein to be translocated to the Golgi bodies, then this glucose needs to be trimmed off. Okay, so Now, this is the precursor oligosaccharide which has three glucose molecules. Then glucose trimming happens. So, this glucose trimming is part of the protein folding complex. Okay. So, once glucose is trimmed, it is actually recognized by the calnexin or the other chaperones. Then it moves to the if it is not trimmed properly or the number of required glucose molecules are still there, then it sends the protein back to have it trimmed and folded. Okay, so here the other chaperones come into play, and that helps in proper folding of this particular protein. Once the protein comes that have only one glucose molecule left on this particular oligosaccharide, this seems to be a perfectly folded protein. Now, this particular calnexin actually sends it to the last glucosidase that trims off the final glucose moiety, and now the protein is fully folded normally, and now it is actually ready to be transported to Golgi bodies. Okay, so the chaperone's work now starts where the oligosaccharide is actually added on the particular protein molecule. Okay, is it fine? Is the explanation fine? So, any doubts until now for the protein folding and how the protein is translated and translocated in the endoplasmic reticulum human? Okay, if not, then we'll move forward. So, tunicamycin is an antibiotic that inhibits the synthesis of carbohydrates. Which of the following organelle activity will be affected by tunicamycin? So, what can be the correct answer? Okay, any more answers? Mhm. Yes, yes. So, the correct answer is option 3, that is endoplasmic reticulum. Okay, because here the oligosaccharide is actually playing an important role because it helps in the proper folding of the protein. Okay, so if we apply tunicamycin, that is an inhibitor of the carbohydrate synthesis, so it will affect the oligosaccharide application to particular protein, and then the protein will not be able to fold properly. Okay, so let's move forward. Now we will be moving to the other compartment, that is Golgi. Okay, so Golgi apparatus is, so what can be the correct answer? Okay, so the correct answer is option 3. So it is actually flattened membranous sacs. Okay, so this is the basic electron micrograph of Golgi apparatus. So this is a, like you can see here, this is actually a various membrane sacs that are compiled on each other. Okay, so this is how it looks in an electron microscope. And it has two sides. First is the cis phase and the trans phase. Okay, so cis phase actually receives the molecules from the ER. Okay, so in the form of vesicle, so it receives it, and the trans phase actually ships it to the outside or to the next particular organelle. So there are two phases, cis phase and the trans phase of the Golgi. So this is cis Golgi network and this is trans Golgi network, TGN. Okay, these particular extensions, or the what we call it, the vesicle forming units of the Golgi are called as cisternae. Okay, so different membranous sacs of the Golgi are called as cisternae. Okay, so any doubts in this particular uh, question? Okay, so let's move forward. Which part of the Golgi apparatus is closest to the endoplasmic reticulum and receives vesicle containing newly synthesized protein? So, what can be the correct answer? Yes, it is the cis Golgi network. Okay, so this is Golgi network that actually receives the protein from the endoplasmic reticulum and it transports it to the trans Golgi network. So, uh, if the endoplasmic reticulum is here, so the cis would face towards the nucleus or away from the nucleus. So, what can be the answer for that?
Towards nucleus.
Okay, anybody else? Okay, anybody else? Okay, so what happens is, because this is the receiving end, this will receive the proteins from the endoplasmic reticulum, and endoplasmic reticulum is actually the extension of the nucleus. Okay, so whenever anything comes from the ER, it will receive from the cis Golgi network. So the cis will be present towards the nucleus only. Okay, not away from the nucleus. The away from the nucleus would be the trans Golgi network, because it has to release the proteins towards the other organelle or the plasma membrane. Okay, so that's why it will be near or towards the plasma membrane basically. Okay, is it clear now? Okay, so let's move forward. What is the role of Golgi apparatus in the cells? So, what can be the correct answer? Okay, any more answers? Hmm. Okay, any more answers? Any more answers? Okay, so the correct answer is option 4, that is processing and secrete sorting of proteins. Okay, so carbohydrate synthesis is like present in every organelle, not specific organelle is there. It majorly happens in cytoplasm. Then lipid metabolism is taken care by the SER. ATP production by mitochondria, and processing and sorting of proteins is majorly done by the Golgi apparatus. Okay, ER only helps in folding of the proteins. But in the ER, because of the presence of SRP, the proteins that need to be present on the membrane or its secretory, it is actually helps in synthesizing those proteins. But once it moves out from the ER to the Golgi, now it is Golgi's responsibility to locate the protein where it is destined to. Okay, for example, if it's secretory, then it will move to the outside. If it's on the plasma membrane, it will be attached to the vesicle that will merge with the plasma membrane. Or if it needs to be present on the lysosome or the endosome, then it will move forward in that way. Or if it needs to be moved to the endoplasmic reticulum. So the sorting of the proteins is basically done by the Golgi apparatus only. Okay, so any doubts in this particular question? So glycosylation starts from the endoplasmic reticulum, and like in two places it occurs, either in the endoplasmic reticulum or in the Golgi apparatus. So glycosylation occurs in these two places. So any more doubts in this particular question? If not, then we'll move forward. Which of the following is not involved in regulation of the Golgi organization? So what can be the correct answer? Okay. Any more answers? Hmm? Any more answers? Okay. So what happens is, in basic terms, I'm explaining now. So the Golgi is also a membranous sac. Okay. So what happens is, there are no chaperones involved in the formation of those membrane structures. Okay. As you see, this is basically a diagram of the Golgi. This is how it is arranged. Okay. And so this is actually a membranous compartment. So it will require the concentration of water to get these particular sized compartments. Okay. So different concentration of the or the hydrophilic environment inside the cell will actually help this particular thing. Okay. So here there will be no role of chaperones here. Chaperones will only help in protein folding, right? How microtubule helps? Microtubules basically help in arranging these particular sacs in a order. Okay, that's how a Golgi is arranged inside a particular cell. So microtubules has a major function. Then the ARF1 is actually a GTPase. Okay, so here the correct answer is chaperones, because here it is not involved actually. Okay, and ARF1 is a GTPase. Okay, so that helps in the Golgi organization and functioning, as well as it helps in membrane trafficking and vesicle formation. Okay, okay, and this CTVP1 or BARS is actually a Golgi localized protein, and along with ARF1, it helps in promoting the membrane fission so that this structure is actually being formed. Okay, into separate compartments. So these proteins actually assist in the formation of these membrane compartments, and these are transporter proteins basically. So they help in promoting the transport of these particular enzymes or the molecules that are present in the Golgi. So they also help in the reorganization of the Golgi bodies along with the microtubules. Okay, so any doubts in this particular question? Okay, if not, then we'll move on to the next one. Which of the following is correct? Root followed by the endocytosed product. So, what can be the correct answer? Okay. Any more answers? Okay. So, first of all, what is actually endocytosis? So, endocytosis is actually entering of any foreign compartment into the cell, and the cell actually accepts it in the form of vesicle or the endosome. Okay, so the vesicle that is formed after the entry of one of the molecules inside the cell is called as an endocytic vesicle. Okay, now this actually moves to first to the endosome, then it will move to the Golgi bodies. From Golgi, it actually moves to the because it also has some receptors that is internalized. Okay, so these Golgi is actually helps in movement of this particular receptor to the plasma membrane. Okay, so it will be from the plasma membrane to the endosome to Golgi. Then through Golgi in the form of vesicles, it will move to the plasma membrane. Okay, so this will be the route of the endocytosed product. So this is option number 3. Okay, so this is a basic diagram of the what I have explained just now. So this is the receptor that will make an entry in of the ligand into the cell. This will be the this will now move to the endosome. Okay, so this is an endosome, and now from this particular endosome, it will either go to lysosome or to Golgi for processing, and from here the receptors will back go back to the plasma membrane. Okay, so this will be the route of a particular ligand that is endocytosed by the cell. Okay, so any doubts in this particular question? Okay, if no doubts, then we'll move on to the next question. So, which of the following best describes the role of vesicles in the endomembrane system? So, what can be the correct answer? Okay, any other answer? Yes, the correct answer is option 3. That means they help in transport molecules between organelle and to the plasma membrane. Okay, so any doubts in this particular question? If not, then we'll move on to the next question. Now we'll be moving to the third component of the endomembrane system, that is lysosomes. Okay, so lysosomes are called suicide bags of the cell because. So what can be the correct answer? Okay, anyone answers? Okay, okay. So the correct answer is option two. That means they carry hydrolytic enzymes which, when released into the cytoplasm, can kill the cell. Okay, so this happens when a cell is actually damaged or diseased. So the body needs to clear those cells, and during clearance, the lysosomes become active and they release these hydrolytic enzymes inside the cell that actually kills the cell. Okay, so this is a basic diagram. It has a membrane. Okay, then it has acid hydrolysis. So the pH of the lysosome is maintained near to 5. So it has a acidic pH in its lumen. This acidic pH is actually required for these hydrolyses to work. Okay, so any of the digested product, any like unwanted product that is present inside the cell is digested or recycled by the lysosomes. Okay, if any of these proteins are actually secreted inside the cytoplasm, will it affect the normal conditions? This is a question to you all. So first, the actually endocytosis depends upon what is the cell wanted to do with that. Okay, so if that is actually being required, that is a signaling molecule. So it will depend upon the molecule that needs to be signaled. Okay, so for example, if it needs to enter the nucleus, it will go through Golgi, then to ER, then to nucleus. So likewise, it will depend upon which of the particular function of that particular ligand is. Okay, so first it will go to endosome, then it will go to Golgi, and from there it will be sorted into where the signal needs to go. Okay, so now my question is here, if any of these enzymes is secreted inside the cytoplasm under normal conditions, so what will happen? Will it affect the cell? Will it not affect the cell? Will it work? Will it not work? What can happen? Okay, how? Yes. So, yes. So, what happens because the lysosome is maintained at a very acidic pH? So, this acidic pH is actually required by these enzymes to function. In the cytosol, it's a physiological pH that is near to 7. So, if any of these enzyme is by mistake secreted into the cytoplasm, it will not work because these enzymes work only at an acidic pH. That is why these are called as acid hydrolases. Okay, so this is the thing, and this acidic environment is actually maintained by a proton transporter on the lysozyme membrane. Okay, so let's move on to the next part. This lysozyme actually, yeah. So because the membrane needs to be maintained because of this acidic pH. Okay, so this lysosomal membrane is meant protected from the acidic hydrolases by lysosome specific expression of membrane proteins, that is LAMP1 and LAMP2. Okay, these proteins are protected from the acid environment by the glycosylation. Okay, so these proteins have a high amount of glycosylated molecules, and then because of the presence of this glycosylation, they resist the digestion because of the acidic environment. Okay, so these lysozymes are actually involved in a suicide of the cell that is called an autophagy. Okay, so autophagy is basically, the meaning is it's self eating. That means when the cell recognizes its components and it's non functional or it's damaged, so to clear it off from the body, an autophagy mechanism actually starts. Okay, so autophagy, what happens in autophagy, in which a cytoplasmic cargo is actually being moved to a double membrane vesicles delivered to the lysosome for its degradation. Okay, so this is a cell, and for example, this is a mitochondria that needs to be removed from this particular cell. Okay, so what will happen? A double membrane sac would be formed near this mitochondria. Okay, so this double membrane sac would be formed, and this sac will now be delivered to the lysosome. Okay, so this is a lysosome. So now this will fuse with the lysosome. This is a mitochondria, and now this lysosome will actually digest this mitochondria. Okay, so this is basically an autophagy process in which the cell only allows to eat its components when it's non-functional or it's damaged. Okay, the substrate of the autophagy include different organelles such as mitochondria, aggregate prone proteins that cause neurodegeneration, and the pathogens. So, if, for example, what happens whenever a cell is infected by a particular pathogen, so in that case also the cell recognizes the affected part, and then the affected pathogen or the pathogen that affected the cell is surrounded by this membrane, and this is directed to the lysosome for its degradation. Okay, and because it's an important discovery, and this is how a cell maintains its dignity and the function that it performs, it's actually a Nobel Prize discovery. So Sir Dubé, Claude, and Pallad are actually being awarded the Nobel Prize for the discovery of an autophagy, okay, in 1974. Right? So any doubts till now? Okay. So now let's move forward to the process of autophagy in simple how it happens. Okay. So there is actually a stimulus first. Okay, so the stimulus is either nutrient scarcity, absence of growth factors, or low oxygen levels. That means nutrient scarcity means starvation. Okay, so there are a range of signals that actually stimulates the process of autophagy. So these are the important signals that actually evokes autophagy. Okay, so these signals are then like introduce the formation of a double layered membrane structure called as phagosome or phagophore. Okay, so this phagophore is dependent upon the type of molecule that needs to be degraded or that needs to be eaten. Okay, so now this size of this phagophore will depend upon the target. Then it will start assembling near to that target and it will engulf that particular target. Okay, so it will surround that particular molecule. For example, here it is the mitochondria, damaged mitochondria. So it will start expanding its size surrounding that particular mitochondria, and it will surround it. Okay, now there are certain proteins that are involved in the formation of this autophagosome. Okay, so these proteins are then recycled to the original function and the original location where they are actually belong, because they only help in the formation of this particular autophagosome. Okay, so now when this autophagosome is formed, this now fuses with the lysosome. Okay, so this is a lysosome, this is a autophagosome, and the fusion, because it's a double membrane, it need the inner membrane needs to be fused. Sorry, it needs to be degraded. So the fusion of the autophagosome with the lysosome, okay, leads to the fusion, the degradation of the inner membrane. Now this uh, complete structure after the formation of the autophagos fusion of the autophagosome and the lysosome is called as an autolysosome. Okay, because now this damaged mitochondria will now be degraded, and the products that are formed from this degradation will be recycled in the cell. Okay, so this is how a basic autophagy functions inside the cell. So any doubts in this particular slide? So keto bodies are basically a undigested form of a lipid in simple terms. Okay, so whenever there is a like when we go for starvation, first there is a sequence of molecules that are degraded. Okay, so when the carbohydrates are the major form of energy, right? So when we go for starvation, the body look for nutrients in the stored forms. So first the carbohydrates are degraded, then come proteins, and then come lipids. Okay, so this lipid molecules, once they start degrading, they lead to the formation of ketone bodies. Okay, so these ketone bodies are basically accumulations. So they are not degraded inside our body. So once these ketone bodies are accumulated, then it's a like bad sign for the body, because now the starvation has like reached to that extent that the lipid molecules are also started degrading inside the body. Okay. So, yes, 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 yes. So that's how, like, for example, if, like, for example, if any politician go for fasting, then, like, this is actually a political stunt. Okay. So, like, uh, go to that extent that they'll not be able to, like, hold that, and the person may die. Then they are asked to actually clear of their fast. Okay, so that's how the, like, like detection of this ketone bodies is done. Okay, any more doubts in this particular slide? uh, it might happen, but till now there are no reports as of now, but it can happen. Okay, because fasting actually, like, in fasting, the stored part of the body, okay, the storage form of all the nutrients inside the body, now starts into the, like, degradation. So the body needs nutrients, and it will utilize those stored parts to like, revive those nutrients. Okay, so that's why it can happen in intermittent fasting also. In case of obesity, actually, the obese condition is different. Okay, there are many factors involved. It is not only linked to lipid degradation. There are many different factors that are involved in obesity. So only taking lipid degradation drugs would, I think, not help at all. Like, it will help, but to some extent. They have to take more medicines for, like, more metabolic, to get the normalcy in all the metabolic processes. Okay, so I think we are shifting our focus to some other thing. So let's move on to the next slide. Yes. So now we'll be moving to the last component of our lecture, that is vacuoles. Okay. So the major component of vacuoles is, so what can be the correct answer? Okay. Yes. The correct answer is water. So the water is the major component of the vacuoles. Hydrolytic enzymes, acid phosphatases are present in the lysosome. Lysosome. And aldolase is present in the cytoplasm. So it's an enzyme basically. Okay, so let's move on to the next question. Which of the following statements about vacuoles is not true? So what can be the correct answer? Okay, any more answers? Okay, any more answers? Okay. Okay, so the correct answer is option 4. That means they do not help in regulation of the cell division or the plant growth. Okay, so basically, vacuoles are only for storage. Okay, they may store water, they may store food, they may store toxic products. Okay, if they store water, for example, in case of the animal cell, okay, vacuoles are not that persistent. If some of the vacuoles are present, they are small sized for the storage only. Okay, in case of plant cells, these are more evident. Okay, because in case of plant cells, they need to store water. Okay, how will Oh, okay. Wait. We'll first discuss this question. So, plant cells basically store water. Okay. Now, what will happen if the plant cells actually store water? They help in turgidity and rigidity to the plant cell. They help in maintaining the osmotic pressure, and likewise, in case of the plant cell, the major plant vacuole actually also stores the toxins that helps in protecting the other parts of the plant. Okay, so it has storage for both nutrients and the waste products, the degradative compartment, and it's actually if the vacuole store water and it increases the water content, then the size of the cell also increases because the size of the vacuole also increases. Okay, then it helps in maintaining the osmotic pressure inside in the plant cells. Okay, because in case of the plant cells, they have a major thing with the water content because they need to transport water from one part of the part to plant to the other. So they need a particular thing in their cell so that they can actually remain in that particular position and in that particular composition. Okay, if that would not happen, then the plant cell would burst easily. So these vacuoles actually help this particular plant to maintain their size and the osmotic pressure as well. Okay, so any doubts in this particular question? Okay, so vacuole also has a membrane, and that membrane is called as tonoplast. So the covering of the vacuole is called as a tonoplast. So any doubts in this particular question? Okay. If not, then we'll move on to the next question. Cell sap is. So what can be the correct answer? Okay. Any more answers? Okay. Any more answers? So the correct answer is option number two. So cell sap is basically water mixed with different nutrients that is basically stored by plant. Okay, so these nutrients are the food formed by the plant and different micronutrients that the plant absorbs. Okay, so this cell sap is basically the mixture of water and all the nutrients that are absorbed or formed by the plant. Okay, so this is the major meaning of the cell sap, and this is cell sap is basically stored in the plant vacuoles. The central vacuole that is present in the plant stores the cell sap. Okay, so any doubts in this particular question? Okay, if not, this was the last question for today. So if you guys have any doubt in any of the concept we discussed today, you can tell. We can discuss this right now. Um, Peroxisomes cannot convert toxins to antitoxins. Actually, the toxins are like the antitoxins are not the converted products of the toxins. Okay, these antitoxins are the compounds that can inhibit toxins. Okay, so I think the toxins can be like peroxisomes help in like what we call like inhibit the function of those toxins. Okay, but they'll not convert it into antitoxins, the peroxisomes. That's what my understanding tells. But I can check and let you know in the next class what is actually like the function. The peroxisomes majorly help in lipid metabolism. Okay, but whether how it handles the toxins, I need to check. Okay.
Sir, actually, as per my studies,
Hmm.
in intermediate level only, peroxisomes, they are the target sites for uh, conversion of toxins into normal forms,
Uh,
that is not antitoxin.
uh,
So, vacuole, I did not study that it may store toxins like that. So,
oh,
that is my inference. regarding my answer.
Okay, okay, okay. Sure. I'll check on that and let you know in the next class.
Okay.
Okay.
Okay, sir. Thank you, sir.
Sure, sure. Okay. So, if no more doubts, then we'll close the session for today. Thank you guys for joining in.
Thank you, sir. Thank you so much.
Yeah, thank you, thank you. Sure. Thank you, thank you.